Hello, this is Nick Cap, and this is going to be an introduction to metabolism in roughly about 20 minutes, and let's see if we can do it. Basically, I try and start this lecture by talking a little bit about thermoelectric materials. I got this really kind of neat stove that I build a fire inside of there, and I get electricity out of it. And so that's a classic example of what we call conversion of energy. That is, how can we take one energy, heat energy, and convert it into electric energy? Life does much the same thing, that it converts kind of chemical bond energy into an energy that we can use. Okay? So basically, since cells are living things, they extract energy from their environment, basically their food, and then they apply that energy to perform work. And again, cells can do lots of work, and so here are photosynthetic, not photosynthetic, but bioluminescent mushrooms. Here's Pacifica. At certain times of the year, we see bioluminescent waves, and our friend the squid that is bioluminescent as well. And so that's kind of interesting. It's electromagnetic radiation from living things. Wonderful. Let's define this. Metabolism is the total of an organism's chemical reactions. It's all the things that goes in there. Again, these reactions to occur have to have a source of energy, and these must be highly regulated, and we need to understand the concept of what we call substrates in our products. A metabolic pathway begins with a specific molecule, typically a substrate, and ends with a product. Our raw materials and our product, and we need to know that. Each step is catalyzed by a specific enzyme, and so again, an enzyme can't do a lot of different things. And so here's a classic metabolic pathway that we have. We have our starting molecule, or starting substrate, is acted upon by enzyme 1, this is reaction 1, and it produces a product. That product then can be the substrate of enzyme 2, that can be the substrate for enzyme 3, and so on and so on and so on. And so even though we start with molecule A and we end up with molecule B that are vastly different from one another, they are made by putting successive enzymes, in this case here a set of 3 enzymes, in order to make that product. Catabolic pathways are pathways that release chemical bond energy by breaking down complex molecules into simpler molecules, like a hydrolysis. Cellular respiration is the breakdown of glucose in the presence of oxygen. An example of that, what we call a catabolism. Anabolic pathways are ones that consume energy and build complex molecules. Bioenergetics is the study of how organisms manage their energy resources, and that's something that we need to do. So, real quickly, what is energy? Energy is the capacity to cause change. Energy exists in various forms: kinetic, heat, potential, chemical energy. These are all energy forms that we need to know. This is a great slide. Here we have a diagram with potential energy, we have kinetic energy, and then climbing up converts kinetic energy into potential energy. And so, again, we have this is a cycle of where this energy can be produced. Thermodynamics is the study of these energy transformations. A closed system is isolated, and an open system where we can see transfer between the systems. And so organisms are, in terms of energy, open systems. According to the first law of thermodynamics, the energy in the universe is constant. Energy can be transferred and transformed, but it cannot be created or destroyed. The first law is called the principle of the conservation of energy. And so energy is always there. The second law of thermodynamics is that energy transfer or transformation increases the entropy, the disorder of the universe. And so again, when we have that, we have an increase in disorder. And so again, here we have the first law of thermodynamics, chemical energy, and the second law of thermodynamics, where we have that more disorder being produced. All living cells unavoidably convert organized forms of energy to heat. Spontaneous processes occur without energy input, and it can happen quickly or they can happen slowly, but they're spontaneous. And for a process to occur without energy input, spontaneous equals energetically favorable, it must increase the entropy of the universe. So that's one of the things that we need to know. So energy flows into an ecosystem in the form of light and exits in the form of heat. And so, again, life can make something ordered like this because there are other parts of the universe that are a little bit more disordered. Why is that not going? Again, the same thing here. Free energy is the energy that can do work when temperature and pressure are uniform, as like in a living cell. And so free energy equals the total energy, delta H, and change in entropy, delta S, and in temperature, Kelvin. And so we're going to need to be able to know a little bit about this energy. Free energy is a measure of the system's instability, its tendency to change to a more stable state. During spontaneous change, the free energy decreases and the stability of the system increases. Okay. Equilibrium is a state of maximum stability. A process is spontaneous and can perform work only when it is moving towards equilibrium. And so again, this is a little bit more of a description of that. Exergonic reactions are proceed without net release and free energy, and it's spontaneous. An endergonic reaction absorbs free energy from its surrounding and is not spontaneous. Okay. And so here what we see here are reactants, here are products, free energy, and then the amount of free energy released to our products, and this amount. So this is an exergonic reaction, this is an endergonic reaction. So we release free energy for an exergonic reaction, and we need to add free energy. And again, this free energy can be used. Defining feature of life is that metabolism is never at equilibrium. Once you reach equilibrium, basically you are dead. Okay? And so it works about this. A cell does three main kinds of work, what we call chemical work, making bonds, breaking bonds, transport, moving things across the membrane, and mechanical, we're actin and myosin, slide past one another. To do work, cells manage energy resources by what they call energy coupling. They use an endergonic process to drive an exergonic process. Most energy coupling is mediated by adenosine triphosphate, that molecule. So you know what adenosine triphosphate is, know what the structure is. Ooh, here's the 2', the 3', the 4', and the 5' carbon. Know what those are. So adenosine triphosphate, 3 phosphates, okay? When that phosphate is released in a hydrolysis reaction, we get a release of energy. And that energy, you need to know this, is 7.3 kilocalories per mole under standard conditions. That's the energy that we're talking about. And so these, how ATP performs work is that whatever work that it needs to do can be done with that amount of energy, that 7.3 kilocalories per mole. So here's an interesting slide that we have here. In order to add another ammonia onto glutamine, so glutamic acid, to make glutamine, that requires a positive 3.4 kilocalories per mole. Okay? ATP gives us a negative 7.3 kilocalories per mole, and so we have net in there a negative 3.9, right? So hey, why can't we just use it twice? It doesn't work. And so it's like walking into a store with a $20 bill and saying, hey, we have this thing here that costs $18, but I don't have any change. And so you just give them the $20 bill, and those $2 are wasted. That's why we see heat. Whenever we see metabolism, whenever we see work being done by the cell, we see the formation of heat. That's that little bit of extra energy that does not get used. And so again, here this is looking at how energy is used in movement by ATP as well as in transfer. ATP is regenerated. Okay, it's not created, it's just regenerated. So we take that phosphate and we stick it back on. How much energy is that going to require? Well, if it releases 7.9, it's probably going to take 7.9 to make it. So to talk about a little bit more, a catalyst is a chemical agent that speeds up the reaction without being consumed by the reaction. An enzyme is a catalytic protein. So not all proteins are enzymes, and not, well, we go on that. And so what we see here is this is a hydrolytic reaction where we have sucrose in the presence of an enzyme, sucrase. ASE means that's an enzyme. We will get back glucose and fructose, and we split the water. This hydrogen goes over here. This hydroxyl group goes over here. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or energy of activation. Activation energy is supplied in the form of heat absorbed by molecules from the surroundings. And so that's why a lot of times if you heat something up, you can see reactions to go. And especially if this reaction releases heat, like this one does here, as long as we get over this hump through this transition state, we will see it. So the delta G has to be less than 0 for it to happen. Enzymatic reactions occur because the enzymes lower the energy of activation and barrier. So enzymes do not affect a change in free energy. Instead, they hasten reactions that would occur eventually. And so here's an enzymatic reaction without an enzyme. Here it is with the enzyme. At body temperature, this is more likely to occur than this would occur. That delta G between what we see in the energy of these substrates in the product has not been changed. So what we see here, the reactant of an enzyme acts on is called an enzyme substrate. The enzyme binds to its substrate, forming an enzyme-substrate complex. The active site is the region on the enzyme where the substrate binds. Induced fit of a substrate brings chemical groups to an active site in position that enhance their ability to catalyze a reaction. And so in this case here, we're showing where actually an enzyme can actually fold around a substrate in order to induce its fit in order to do it. The active site can lower the energy of activation barrier by orienting substrates correctly, straining substrate bonds, providing a favorable microenvironment, covalently bonding to the substrate. All these things can happen. And so it's almost like a lock and key. We see that enzyme-substrate complex, and then we see that change occurring. An enzyme's activity can be affected by the pH and kind of a number of different things. Anything that can affect the shape of a molecule of the enzyme can affect its reaction. And so what we see here is that certain enzymes will have optimum temperatures and optimum pHs, and we understand that. Cofactors are non-protein enzyme helpers. Cofactors may be inorganic, such as metal in ionic form, like zinc is an example, or organic. Organic factors are called coenzymes. Coenzymes include our vitamins. It's one of the reasons why we need vitamins. It's one of the reasons why we need some of the micronutrients that we have. Again, an enzyme can have an inhibitor. A competitive inhibitor is one that looks a lot like the substrate, and so it will compete with the substrate for the active site on an enzyme. A non-competitive inhibitor is one that can bind somewhere else on the enzyme and stop its function. So a lot of the drugs that we want, you want them to be non-competitive inhibitors. And so here's an example of that non-competitive inhibitor over here. And so even though this is the active site over there, and even though that inhibitor binds on the exact opposite side of the protein, it can affect its shape of that particular active site, and it will no longer bind. Okay, and so here's an example of a topic that we call site-directed mutagenesis, where we can actually take an enzyme and we can evolve it. And believe it or not, all of life may have evolved from one enzyme. It's a theory. Enzymes are proteins that are coded by genes. Changes or mutations in the genes lead to changes in the amino acid composition of an enzyme. Altered amino acids in enzymes may result in novel enzyme activity or altered substrate specificity. Under new environmental conditions, a novel form of the enzyme might be favored. Again, chemical chaos would result if a cell's metabolic pathways were not tightly regulated. A cell does this by switching on or off the genes that encode specific enzymes or by regulating the activities of the enzymes. Allosteric interaction regulation may either inhibit or stimulate an enzyme's activities. And we talked about this. Cooperativity is kind of important. Feedback inhibition is when the end product of a metabolic pathway shuts down that pathway. And so here we have that metabolic pathway. And this is kind of really interesting. And I saw this in graduate school, and it creeped the hell out of me. But threonine is an amino acid that can be actually converted into isoleucine. When there's lots of isoleucine present, it will actually prevent this enzyme threonine deaminase from working. And so what's actually happens if you have actually lots of isoleucine around, it will actually starve the cell for threonine. Again, these enzymes are located in certain structures of the cells. Remember what a cell kind of looks like. And if we have a pathway, if we have enzyme A, and then we have enzyme B, we have enzyme C, and we have enzyme D. If they are all in a linear pathway, they are able to, basically, the substrate is one, but is able to be the product of another.